Anyway, I gotta make the gang go. I do things major. You're irrelevant. Pager. I'm a maverick.
Sorry.
3 is essential. Stay gassed up.
Hello there. Oh, come on. Get a bit closer. Okay, not that close. Back a bit. Oh, for goodness sake, move back! Oh, finally. Yes, I'm losing count of how many installments I've done of this now, but I think it's the 7th. But yes, hello. Welcome back. Oh. Whew. It's tiring work, this, you know. Now, last time we talked about expressing genes in fungi. Well, particularly yeast. So we're going to carry on going, well, onwards and upwards, you might say, you might not say, and look at expression in animals today. Later in the video we'll be talking about transgenesis, that's transforming a whole organism, a whole animal, so every cell has the gene that you want. But first we're going to be talking about transforming animal cells in tissue culture. First of all, why do we need to express genes in animals? Well, animals tend to be top-notch when it comes to post-translational modifications, which we talked about in the last video. So if you want the full extent of translational modifications to produce your protein, then animals are the one for you, really. Now, in particular today, we're going to be talking about mammalian expression vectors, because that's the type of expression vector which we most commonly use in life. Now, there are very common cell lines which we use when expressing genes in animal cells. One of them is CHO, which is a Chinese hamster ovaries. That's a very common one. Don't ask me why, it sounds a bit random to me. And here is an example of a mammalian expression vector. Now, as usual, we'll go through them. It's an episomal vector, so just like the shuttle vectors we talked about in the fungi video last time. So it's not integrated into the mammalian chromosome at all, it just hangs around inside the cell. So what do we have? There are loads of things here which are not new to us whatsoever. We've got the origin of replication through E. coli. We've also got the BLA gene for antibiotic resistance in E. coli, that's not new. We've also got our multiple cloning site where we insert our DNA. That's also not new, well that's what we're doing this for in the first place really. But we've got a lot of other things which aren't that familiar to us. So we've got this NEO, that codes for neomycin phosphatase, and that's a neomycin resistance gene. So that's the selectable marker in animal cells. So the BLA gene is for a selectable marker in E. coli, and that's the selectable marker in animal cells. Now there's also this, the SV40 origin of replication. Now that, just like the OEE replication for replication in E. coli here, that allows for replication in the mammalian cells. Now SV stands for simian virus. This is a monkey virus, so it's able to replicate within these mammalian cells, so it's very useful. Now either side of where you've got your insert, you've got other things here. Now at the end, we've got the PA, that's from polyadenylation signal, that adds the string of A bases onto the end of your mRNA to increase its stability, so to increase its chance of successful translation. We've done that before. And here we have the promoter upstream of the insert, and that is the CMV promoter. That stands for cytomegalovirus. Now this cytomegalovirus is useful because it has a high rate of transcription, so it produces loads of mRNA which will contain your insert, so that's a very useful promoter to have. So all you need to do is first replicate this plasmid in E. coli, that's what this bit's for, and then introduce it into your tissue culture, let's say your Chinese hamster ovary cells, and then it will continue replicating and producing the gene that you want, and you can extract it, and all happy days. So there isn't really that much new here, it all seems to be as expected. The only thing that's changed is the different selectable markers for use in animals, and the different promoters and origins of replication. The rest of this is basically the same concept that we talked about, especially last time. What about transgenesis? That involves transforming a whole organism. Now, the most common animal which we usually make transgenic is the mouse. Just because it's easily kept, it's easily worked with, manipulated, has a high reproductive rate. So it ticks all the boxes really. It's the, what you could say, it's the E. coli of the animal world when it comes to molecular genetics. So, how do you turn your pet mouse into a mouse which produces a particular protein that you want to collect? Well, if you have a pet mouse and you have some highly sophisticated molecular equipment with you, then this is how you do it. One of the problems with transforming animals is that they have something called a germ line. In the very early stages of embryonic development, a group of cells will be given a future for producing the germ line, the ones that produce the gametes, so the sperm and egg cells. So you've got your germ line, and everything else is what we call somatic cells, ones that aren't involved in the production of gametes. So it is very, very important for you to, when you inject your insert DNA, for it to go into the germ line. Because otherwise it won't be passed on to the next generation. You'll just have one transgenic mouse. Now that isn't much use to it. You want the progeny of that mouse to contain the gene that you've inserted, and so on and so on. So you get loads of mice producing the gene that you want. Because let's be honest, no offence to the mouse, but it isn't going to do much good on its own. So therefore you've got to insert your DNA before even the zygote properly forms. So here we have at the bottom is a beautiful drawing once again, is a fertilised mouse egg, right? But as you can see, the two haploid nuclei haven't yet fused together. So you've got a nucleus from the egg and one from the sperm. Now what we're going to do here is extract this egg and through micro-injection insert the DNA that we want to be inserted into one of the nuclei, one of the pronuclei. So then, after that, hopefully, fingers crossed, the DNA that we've inserted will be integrated into that pronuclei chromosome. Around 10 to 30% of the time, this will happen, so it's relatively okay. So then what will happen once you've done that, you'll allow the nuclei to fuse and this zygote to divide and produce an embryo. So then, all the cells in that embryo will contain that piece of DNA that you want because they've all come from this fertilised mouse egg here. So you insert this embryo into a surrogate mother mouse and the offspring produced will contain the gene that you want integrated into all of its cells. But the problem is, each one of those cells is what we call hemizygous. Now this is kind of the same to heterozygous. Heterozygous is when, in a diploid organism, you have two different alleles on each homologous chromosome, right? Well, in this case, one of the chromosomes has this bit of DNA that you've inserted, and the other one doesn't. So it's not quite the same as heterozygous, so we call it hemizygous. Now, that isn't any good. What we need is a true breeding copy. A mouse which is homozygous. And for that, we just do a little bit of simple Mendelian genetics. So you've got to love a bit of simple Mendelian genetics, so let's turn the board over and do so. So, on one of the chromosomes of this mouse is the normal wild type chromosome with no extra DNA inserted. We'll give that a W, right? On one of the other chromosomes, it's got that little bit of DNA that we've stuck inside there. Very sneaky. So we'll give that a lowercase w. Now what we're going to cross that, we're going to back cross that with the parents of that mouse. So we've got a little Punnett square here, and the parents of that mouse are both going to be wild type. So on both of their chromosomes they'll have the normal chromosome with no DNA integrated that we've inserted. So the progeny of that mouse will be this. So as you can see, half of the offspring have the chromosome with the DNA that we've inserted in it, so 50%. So once we've selected ones which do have the integrated DNA from the ones that don't, what we then have is a nice group of mice which have the DNA that we want in the same place on the chromosome. The DNA that we've inserted could have integrated itself anywhere. So that's why we do this crossing back with the parents thing. So then what we do is do a sibling mating. So we take two of those offspring, well, preferably a male and female, otherwise this would not work. And then you mate them together. As so, we have another Punnett square, and you get this. So what have we got here? Well, here we've got a classic 3:2:1 Mendelian ratio. We've got 1/4 being the wild type, with both wild type chromosomes there. We've got 2 that are hemizygous here, but this is what we really want. We've got a homozygous mouse which on both chromosomes has the DNA that we've integrated. So this is the one that we want. We can use the 2 hemizygous ones for more sibling matings for us to create more homozygous mice. So they're still useful. But this is le creme de la... what's it called? So, that's one way of making a transgenic mouse. But a technique which is more commonly used nowadays, I think, is using embryonic stem cells. Now, embryonic stem cells are cells which are produced in the early embryo which have no particular function. They're undifferentiated. So that means they have the potential to give rise to all possible cell types. And that makes them really useful. Now this is also one of my best diagrams. Admittedly, it's basically been copied from a lecture slide, but I think I did a pretty good job. Thank you very much. In early embryo development, if you do any development biology out there at all, if you know any, well, you should know this, that as the embryo develops, the zygote divides through cleavage, and eventually you get this structure called a blastocyst. So what you've got is a mouse blastocyst. On the other side of the laboratory, you've got a culture of mouse embryonic stem cells, which we are going to insert some foreign DNA into. And we're going to do that through microinjection, just like how we did before. So then this DNA becomes integrated into these mouse embryonic stem cells. Now what we're going to do is introduce these embryonic stem cells to the blastocyst, right in this hole here. So that blastocyst will carry on to divide, the embryo will carry on to develop, and then we implant that into a surrogate mother mouse, once again. Now what we get are what we call chimeric heterozygous mice. Shit, I've just made a mistake. That says heterozygous, it should be hemizygous. Wait a minute. What do we mean by chimeric? Well, that means that the mice that are born are made up of a mixture of these cells from the original embryo and these cells which we've inserted into the blastocyst. So you've got your hemizygous mice, and then basically you do the same back-crossing Mendelian genetics fun that we did before. So eventually, after all that, we can get our homozygous recombinant mice. This is quite useful and quite a simple technique, really. It's also quite useful in working out gene function in mammals, which is obviously very useful in medicine, for example, today. So what we could do is mutate a particular gene on these embryonic stem cells, then insert these cells into a blastocyst, implant them to a surrogate mother, and see what happens to the progeny that's produced. Any effects that the removal of this gene has on that mice will give us an idea on its function. Not so very good for the mouse, of course, because if we knock out a gene which is particularly useful for its living, then it's not going to be a very happy mouse. Now once we've got our chimeric hemizygous mice, we can work out which of these progeny had these embryonic stem cells incorporated within the germ line. So the ones that do have the gene incorporated in the germ lines are the ones we're going to keep, because they're the ones that can pass it on to future generations. And that is it! Everything you wanted to know to make a transgenic mouse. I advise you to go up there, find a pet mouse, and do this all yourself. Actually, no, I don't. No, that would be horrible. Yeah, poor mouse, you don't know what you're doing. You know, test it on your... I don't know, test it on your gran or something. I don't know. For the moment, goodbye. I'm meant to... It's okay. That way, okay.